Brain development and mental health are significantly influenced by early childhood environments, with some being genetically predisposed to psychological vulnerability, as shown by Caspi et al. And Kim Cohen et al. Halligan and team has shown that children whose mothers are depressed are more likely to develop psychological problems compared to children with non-depressed mothers. And Wiseman et al. Found that early intervention of children with depressed mothers can decrease the likelihood of developing psychological disorders. Studies done on rodents have similar findings. Tang et al. Showed that short exposure to new environments during the early postnatal stage decreased anxiety-like behavior and improved the hypothalamic pituitary adrenal stress response. Together, these studies illuminate the interaction between how genetic susceptibility and environment both play a role in shaping neurodevelopment, emotional, and behavioral health. High and low response to novelty are phenotypes present early on in rats' life and are associated with distinct hippocampal development. Previous studies showed that these heritable phenotypes can also be modified by environmental factors. We also know from previous work that high response and low response mothers have distinct maternal styles. Coupled with the significant relationship between maternal style, neurodevelopment, and the hypothalamic pituitary adrenal axis, our study examined how maternal behavior affected brain development and behavior in the high anxiety, low response offspring. We focus on low response offspring because previous work has shown that high response offspring are resistant to environmental manipulation. For this study, we developed a rat model of different emotional differences in responses to novelty by selectively breeding Sprague-Dawley rats. Rats bred for high response to novelty, denoted BHR, showed strong exploration, greater risk-taking, impulsivity, aggression and tendency towards addictive behavior. Rats bred with low response to novelty, denoted BLR, showed high levels of spontaneous anxiety and depressive-like behavior. These phenotypic differences allowed us to foretell whether the rat contained the high response or low response traits from birth and throughout the rat's life. We used a cross-fostering paradigm in which low-response offspring were randomly assigned to being cross-fostered by low-response mothers, cross-fostered and raised by high-response mothers, or raised by their biological low-response mothers, which was our control group. We also included a high-response control group where high-response offspring were raised by high-response mothers. The study included 2 separate experiments and only male offspring were studied. The first experiment consisted of behavioral tests to examine the impact of cross-fostering on behavior and social interaction of the anxiety-prone, low-response adult males. Tests included an open-field test, an elevated plasmase test, a novelty-suppressed feeding test, a social interaction test, and a forced swim test. The second experiment consisted of a second group of rats where the cross-fostering procedure used for the first experiment was repeated. Tissue collection, RNA extraction, and genome-wide expression profiling was conducted. In the open field test, we found that the high-response control rats spent more time in the center of the box, had greater activity, and spent less time grooming than all the other low-response groups. Cross-fostering subtly decreased anxiety-like behavior of low-response offspring, and low-response offspring raised by high-response mothers showed increased locomotion and less grooming compared to other low-response groups. In general, low-response rats fostered by high-response mothers differed than the other low-response rat groups. The elevated plasmase showed similar results to the open field test, with the high-response control rats spending more time in the open arms, showing less stress-induced defecation and greater activity than the low-response groups. Again, cross-fostering subtly decreased anxiety-like behavior, with the low-response rats being raised by high-response mothers, showing less stress-induced defecation than the other low-response rat groups. In the social interaction test, the high-response control rats showed a high level of interaction with other rats compared to the low-response rats. Cross-fostered low-response rats with high-response mothers showed increased interactions with other rats compared to the other low-response groups. The high-response controls showed the least stress-induced defecation. The low-response rats fostered by high-response mothers also showed significantly less defecation than the other low-response groups. In the novelty-suppressed feeding test, high-response control rats showed the shortest latency to enter the food zone, and least time grooming compared to the low response groups. All cross-fostered low-response groups showed significantly less time grooming than the low-response control group. While the high-response control group spent the least amount of time immobile in the water, all other low-response groups showed a similar level of immobility, and so it was determined that cross-fostering did not impact depression-like behavior in this test. Gene expression analysis of the cross-fostered low-response rats with high-response mothers showed significant developmental gene expression within the amygdala compared to the two low-response groups, and found few differences in gene expression when comparing the low-response control and the low-response cross-fostered groups. When comparing groups at different life stages, cross-fostering had little effect on amygdalar gene expression until 21 days old, with the majority of the altered genes being upregulated in the low-response offspring with high-response mothers. It is important to note that gene expression changes were most pronounced at 75 days old, and these changes were specifically triggered by cross-fostering the low-response offspring with high-response mothers. We performed a partitional cluster analysis in which we examined amygdala gene expression by grouping genes altered by cross-fostering. We chose to focus on 75-day-old rats since that was the age when the greatest numbers of genes were altered in the amygdala. 103 genes were found in the amygdala dataset of 75-day-old rats and clustered into 4 groups based on the slope of the gene's expression pattern over time. We chose several transcripts in cluster 1 that were related to learning and memory and used real-time PCR to confirm that 6 genes were upregulated in the amygdala of the cross-fostered low-response rats. 2 of the 4 gene clusters were altered by cross-fostering and were associated with learning, memory, and vesicle-mediated transport. A pathway analysis was also performed on the 75-day-old rat amygdala dataset, which revealed 123 pathways that were significantly altered by cross-fostering. This table shows the top 10 pathways. Note that many pathways involve genes related to synaptic signaling, glutamatergic signaling, adrenergic signaling, and apoptosis. In general, the same genes were not altered across different ages, suggesting different paths are engaged at different stages of life. So, what do these findings mean? In an earlier study, we found that low-response rats had a baseline difference in hippocampal development compared to high-response rats, and that we could improve low-response phenotypes in hippocampal development in adults with an early life injection of growth factor 2. However, it was surprising that in this study we found no genes were found to be expressed differently in the hippocampus at any age. Yet we were able to improve aspects of anxiety-like behavior through cross-fostering. This suggests that some aspects of the low-response rats could be related to the hippocampus, while others related to the amygdala. Much work has been done by Meany et al., who show that long Evans rats raised by mothers who display insufficient active maternal care, such as grooming, displayed greater anxiety-like behavior compared to rats raised by mothers who showed higher active maternal care. Our study showed that the low-response mothers exhibited more active maternal care compared to high-response rats, yet offspring of the low-response mothers displayed high levels of anxiety and depression. Meany's conflicting results suggest that the impact of maternal care on its offspring is dependent on the type of rat strain. In summary, by previously showing that high-response and low-response mothers have distinctive phenotypes and maternal styles, we can now see that cross-fostering anxious and depression-prone offspring to mothers who display high-response phenotypes can lead to changes in the amygdala that persist through adulthood, thereby increasing social interaction and reducing anxiety-like behavior associated with low-response traits. This study highlights how early life manipulation, such as cross-fostering, can impact the offspring's adult behavior through neurodevelopment pathways that includes the amygdala, and interestingly also finding no change in the hippocampus. Future work will focus on the molecular and cellular mechanisms within the amygdala that contribute to the low-response, high-response traits. An area of improvement or perhaps future research would be to conduct these experiments on female offspring as well. Since there is almost an equal proportion of men and women in the world, I argue that there is equal importance to learn about female neurophysiology and emotional and behavioral health. Another point to consider is while it is logical to focus on the rat amygdala as an adult, when the greatest numbers of genes were altered in the amygdala, there may be important information that can be extrapolated upon before the rat becomes an adult.